So, okay, we can continue with our scientific presentation for today. So, Nikita, please, you are welcome to start.
Yeah, hello everyone. My name is Nikita. As Katya told, I finished my master's at Tel Aviv University, and now I'm doing PhD in Tel Aviv University in Department of Biomedical Engineering in Momentum Lab. Today's talk topic is chemical exchange saturation transfer magnetic resonance fingerprinting protocol optimization. So I will talk a bit about theoretical background on CEST with examples, about CEST MRF with examples. We'll tell why we need some optimization, and we'll show some preliminary results, which I will present also at the ISMRM this year. So I suppose that most of you already familiar with classical MRI. Let's consider what's the difference between classical MRI and CEST MRI in context of treatment monitoring. And because all my work is performed in the context of treatment monitoring. So the classical conventional MRI is based on the water protons. So on the diagram, you can see that usually we have like two main parts of conventional MRI is conventional MRI and advanced MRI. The first one is kind of T1 weighted images, T2 weighted images, inverse recovery of weighted images. And when we are looking at some cancers like tumors in the brain, we can measure some diameter, cell volume of these tumors. And also we have advanced techniques which measure which are based on diffusion and perfusion, and we can measure cellularity or vascularity of the tumor. And if you will look at some standard protocol for the doctors how to monitor treatment, you will see that it always requires contrast agent injection like gadolinium, but it has some toxic effects. It accumulates in the liver and it's not favorable in a lot of cases. And also another drawback is impossibility or late stage response or pseudo response detection. You can see at the diagram here that for advanced it takes kind of weeks and for conventional it's months, because we can monitor in one case tissue microstructure, in another case tissue macrostructure. And also this information which we'll get from the conventional MRI is non-specific to our treatment. So this changes what we observing. It can be caused by a number of factors. While on the opposite, the CEST MRI targets not the water protons, it targets the protons inside some molecules which can change their saturation with water proton. It's some mobile proteins and metabolites. So our information now is molecular. And this allows to us early stage response detection. We can easily differentiate between pseudo response and real response because we can monitor metabolism, because CEST is sensitive to the pH, and pH, this is like it's molecular information about molecular level of the tissues and tumors. We can see metabolite concentration and protein expression. We can track cells, we can track their death and migration. So we can obtain treatment specific information. So if we know what treatment we used, we can actually see how it works. So it's more favorable, and all changes can be observed even at some days after treatment stop, and the level of information is cells and molecules. How it works? In the CEST, we have two pools. One is the large pool of water and small solute pool. Usually it's some groups like amide, and hydroxy groups, which has proton which can exchange their saturation through the chemical exchange. And we saturate exactly this proton at this particular frequency. For example, here you can see we irradiate at the frequency 8.25. And after that, this proton exchange their saturation to the water. And when it's transfer its saturation to the water, it becomes again unsaturated. After it again saturated and transferred to the another water hydrogen proton, and it repeats over and over again. So signal is amplified multiple times, and this is why this method is more sensitive than, for example, magnetic resonance spectroscopy. So what we are measuring here, here you can see this kind of spectrum data, and this is H1 spectrum. So actually we just measure our water signal, because when the solute proton transfers its saturation to the water, it decreases net magnetization of the water and decreased signal, which we see. So after that, people usually normalize this spectrum by the signal of the water. So we can see here, like the spectrum show us a loss in the water signal value. So at the 3.5 ppm, here we can see the resonance of solute of interest, and at 0 it's water signal. So we saturated water, it's like direct saturation, so we don't see any signal. And after that, you also can see that this spectrum, called this spectrum or CEST spectrum, is symmetrical. So to obtain the kind of pure signal of our pool of interest, we can just subtract the right part from the left part, and we will get the thing called MTR asymmetry, which is magnetization transfer ratio asymmetry. And this is what we actually measure in the CEST. This MTR is correlates with the concentration and pH and other things I told before. Also, we can extract quantitative parameters. It's kind of T1 and T2 in the common MRI, but here it's concentration of our pool and its exchange rate. The first is intuitively, you can understand, it's concentration of some metabolite, and second is exchange rate. It's how often it transfers its magnetization to the water, and usually it is proportional to the pH or temperature, and gives to us a lot of information. And it works similar to common MRI. We have some model. We can create some dependency of our MTR asymmetry from some parameter, for example, this irradiation time. So it's time how long we saturate our pool proton, and we can perform model fitting and extract this information. And there are multiple models, which are most of them based on the steady state solutions of the Bloch-McConnell equations. It's similar to the simple Bloch equation, but it's extended to the chemical exchange. So yeah, after model fitting, we will quantify the exact parameters. So let's look at some examples. The first one is corresponding to the PMID imaging, PMID proton transfer weighted images. Here's one of the first works in the APT weighted imaging. Here you can see 3 sets of images. First is a T1 weighted gadolinium enhanced images, so it's common MRI, and APT weighted images. And the first, you can see in all of cases, we see increased signal intensity. So we suspect that all of them are tumor. But on the APT, you can see that in this area, on a APT, we can see the decrease in signal, while in other 2 cases, we see increase in signal. And after this is mice model, so after that, researchers just took histology of these particular tissues and checked that, yeah, this is actually radiation necrosis. So it was, yeah, I didn't know that this was mice models of the tumor. They were treated with radiotherapy. And yeah, after that, they found histologically that here is a real necrosis and here are 2 tumors. Why it happens? It happens because when we have necrosis, this is cell death, and there is absence of mobile proteins, so we see decrease in APT weighted signal. If the tumor active, it's actively proliferates, the increased content of the mobile proteins, so we see increase in signal. That's simple. So yeah. The next example is amine imaging. Amine, maybe heard about amino acids. So amine is the most sensitive to the pH, to the acidity of the environment. So here we can see examples of the, again, we have two sets of images. One of the is suspension to the tumor and also suspension to the tumor, because on the common MRI images, we can see increase in the signal on the here on the inversion recovery in the both cases. But on the amine weighted or pH weighted, we can see that here we observe decrease in pH, while here it's a normal pH. And there is a hallmark of cancer that when there is cancer, when it's active, the environment are acidic because it needs a lot of glucose, etc., etc. So we can monitor this. And you already can see that this is human data. After that, people with real tumors will they got some treatment, and some of them got the recurrences of the tumors. And already after their death, they took the histology of some regions of the brain and found out that here you can see 3 points, blue, yellow, and red. On the yellow, in all cases, we see increase in the normal MRI and the common MRI, but on the pH weighted, in the yellow, we see some moderate pH, and in the blue, the normal pH, and the red, we can see low pH. So it's red, yeah. And after that, histologically, we confirmed that yes, in the yellow, there is moderate cellularity, there is some necrosis plus tumor cells. In the blue, we can see that treatment worked, but in the red, there is a lot of tumor cells. And the last example is the AutoCEST, which actually hydroxy group imaging. And I put here example of a cartilage, because several years ago, cartilage was something of interest in the physics department. So you can see that here are two images of the healthy volunteer and patient with osteoarthritis. And it was shown that when patient has osteoarthritis, it has decreased content of glucosaminoglican. And we can monitor it with CEST. And here you can see that in healthy volunteer, the MTR AutoCEST signal is increased, while in the patient it decreased. It was just one of the feasibility studies. There are also a lot of different studies which show that the method can be a good alternative to the current gold standard. Okay, let's look at some challenges with CEST. The first is we need to pre-saturate our protons in our pools, and usually it requires a lot of radio frequency power. So there is always problems with specific absorption rate. And like as I showed, there are several models for quantification, some of them, and they based on the steady state solutions. So we need first reach some steady state. It takes some time and a lot of energy. In the clinical case, and this is performed with continuous wave saturation, so just one big pulse, while in the clinics, we want to use something for the pulse saturation. And the next problem is that it's very sensitive to the inhomogeneities of B0 fields and B1 fields and water T1 effects. Here we can see example of two Z spectrums taken from gray matter. So we expect they will have minimal differences, but just uncorrected data, they absolutely different. There is some bias in water in the zero, and correction of B0 makes them more similar, and B0 plus B1 makes them almost identical. So it's very important points to consider. Another problem is that CEST more effective at high fields, especially for the low exchange rate modes. Here you can see numerical simulations of the Z spectrum. And for example, 3 ppm resonance is visible only at ultra high field if we have some small exchange rate, kind of 30 Hertz, and also some simulation of the meteor asymmetry and dependency from exchange rate. So if we have some low field like 3, we can see only such metabolites which has relatively high exchange rate. Another problem is that all the examples I showed you, they based on the qualitative imaging, so just MTR symmetry without quantification. But in reality, this data depends on several factors. For example, on the image acquisition parameter like TR, Philip angle, etc. And also there is contamination from other pools. Like, for example, here you can see example of pool of the spectrum, and there not only one pool, there is also a MID, a MINE, also an OE makes some effect on this asymmetrical. It can be observed only on the right side, so it's hard to disentangle from them. And if we are using just qualitative analysis, that's our interpretation will be biased. So one of the solutions is to use quantitative imaging, but as I told before, all models are based on the steady state solutions. It takes a lot of time. And also, while in qualitative image, we need just to obtain, for example, 2 points. If you need MID, we just obtain on this side of the spectrum and this side of the spectrum, only 2 points. We can extract them, and this is our qualitative image. While in the quantitative image, we need to acquire all the spectrum, because if we want some accurate quantification, we want delete all contaminations. So we need to perform some fitting, which also takes a lot of time. So both acquisition time and reconstruction time takes a lot of time for quantitative imaging. Because if we want to disentangle different spectrums, we need to perform fitting to Lorentzians, like to the sum of Lorentzians. It can take a lot of time, and there are also other methods, but yeah, so that's a challenge. And as a solution for the quantitative imaging, fingerprinting can be a solution. What is magnetic resonance fingerprinting? The goal of this methodology is create the dictionary of signal trajectories, which are unique response to the unique pulse sequences. What does it mean? Since we have block Markov equations, we can solve them. At least we can solve them numerically. And this is actually gives to us simulator of our CEST MRI. So we can put some tissue parameters like T1 of the water, T2 of the water, T1 of solution, etc. We can put parameters of our pulse saturation sequence, like power B1 of saturation, time of recovery. We can put some parameters of readout to the simulator. It gives to us some signal intensity, just magnetization at the end of such pulse sequence. And if we're varying some of these parameters, for example, B1 or T saturation, we will get vector of such intensities, which called trajectory, which our unique pulse sequence, unique response to our unique pulse sequence and in magnetic resonance fingerprinting. So in the CEST MRF, usually people varying only parameters of the pulse sequence. Like here, we have a vector of B1, vector of time of saturation and frequency of saturation, and readout usually is fixed. We can put them to our simulator, and we can create sequence file for the actual scanner. And after that, we can simulate this dictionary. So here you can see set of the signals corresponding to the set of the parameters. And by the way, like usually we use in this research, we use PulseSeq library. It's a library which allows to you program your sequences for the MRI. After that, when we simulated the dictionary and .seq file created by PulseSeq library, we can go to the scanner and scan real data. So we will acquire some set of the images, and if you plot the pixel-wise the signal, we will see some trajectory. And now we need just find the corresponding signal in our dictionary to see which parameters correspond to this signal. Usually perform using some algorithm like dot product matching. So just correlation-based algorithm, or some neural networks. So performing this algorithm pixel-wise, we can obtain several quantitative maps at one time. Like here we can see the exchange rate and concentration of semi-solid macromolecules and amide. The same for the amide. Let's consider some examples. This is one of the first works in CEST MR-F. They had two sequences with varying B1 saturation power, and they performed phantom experiment and mice experiment. You can see that exchange rate, which proportional to the pH, in this case, must be the same. And they observed that this is, as it must be, the same for the concentration, 25, 50, 100, and another phantom. So there is feasibility even with varying only one parameter. There is another example in context of treatment monitoring. The research showing that fingerprinting was successfully applied to the monitoring of oncolytic viral therapy, and it just proportional to the cell apoptosis. So there is a decrease in signal, was histologically confirmed apoptosis. And just another example how multiple quantitative maps can be obtained at once, like here T1, T2, and CEST parameters. Which are challenges with CEST MR-F is the following. The first is that still to be clinical relevant, it must not increase the overall routine scan time, because usually doctors scan T1, T2, some inversion recovery, and if our sequence is too long, everything will be too long. And also SAR problem still relevant. So we want to decrease schedule length. Like for example, here in the previous example, they used such sequence length of 30 with duty cycle, pulse sequence duty cycle 0.5, while we can decrease and get the same efficiency of saturation with increased power of saturation, which is actually not good, or with increased duty cycle. And overall, it will take less time. Another problem is that signals in the dictionary must be distinguishable as much as possible. So if we have big dictionary like 30, not big dictionary, the long sequence, like 30 images in the sequence, that's good. But if you want to make it shorter, that's a problem. And also there is some other problems related to the dot product quantification. That's a bit side thing, but you can read it. Okay, which metrics to measure discrimination ability are used? It's the following. The first is of diagonal Frobenius norm dot product, and the same but for Euclidean distance. They showed efficiency for the classical MRF, not CEST MRF, and also in some pioneer works in the CEST MRF. And the third metric is Monte Carlo simulation of noise propagation. So just we can create a dictionary, we can add multiple times noise and perform quantification and calculate the error. And here are some results of first work in this topic. Here is just an example how different parameters like B1 or T saturation depends on this discrimination ability metrics. And you can see that there is no one solution. This is just waste space for the optimization. It is impossible to find manually. And in this research, they tried just to decrease the length of the sequence and found out that 11 iteration is something what can be done without a lot of heart. And the most relevant metric was Euclidean distance. The state of the art in such optimization is the following. Like, actually, we have analytical solution of Bloch-McConnell equations, and we can put them to some autograph framework like PyTorch and optimize it. This is called AutoCEST. Here the diagram of this model. It consists of the saturation block, which is actually analytical solutions of block-McConnell equations. And the next block is readout block. This is analytical solution of the block equation with rotation matrix. I think you know this thing. You did some laboratory works or something. And the last block is a neural network for the reconstruction, just to overcome problems of the dot product. And this can be done end-to-end. It at once gives to us optimized sequence and optimized network for reconstruction. So here you can see the graphs of this calculation. They are pretty complicated. So overall, this method gives us the schedules with range 9. It seems that's okay. That's all we found the best way how to do that. But the problem is that these analytical solutions only exist for the continuous wave case, which are not clinically relevant. So we cannot use such solution for the real case scenarios. So the further analysis is required, and there was shown that the metric called Kramer-Rao bond is successfully applied in the classical MRF, and it was shown that even in CEST MRF, it reflects the discrimination ability. So I propose try to use it. So this bound is telling to us that covariance of the estimation of some parameter is bounded by the information matrix, so which is actually just squared Jacobian. We can calculate Jacobian in our dictionary just numerically, and we can add to this some classical numerical algorithm for optimization, and thus there is no need of analytical solutions. And here is some examples on LRG9 phantoms with length of the sequence 4. You can see the upper row with images before, using images using initial sequences without optimization, and the lower row is after optimization. You can see in the white ground truth values, and you can see how initially it was pegged to some value, to the highest value, and after it becomes, as it must be, equally between all whiles. The same for the pH and for the other phantom. Here you can see statistics that there is statistical significant decrease in the mean absolute error estimation of the concentration, and also decrease in the estimation of the exchange rate, but not statistically significant. Also, I performed the same experiment on the clinical scanner, so it wasn't so smooth as a preclinical one because I encountered with problems of field inhomogeneity, and I tried to correct it. But overall, I got statistically significant decrease in the concentration, but a real mess with the exchange rate. And I figured out what's going on there. Actually, they're not only a field problem, there are also problem of the spin history, that I was scanning a lot of sequences one by one, and they all affect to each other. And so yeah, I figuring out what's going on there. And also there is really promising results with the healthy volunteer data on the MT pool. You can see here initial images with range 4 and range 8. Just visually, you can see how non-structured and noisy images become more structured and more and less noisy. And like, it's really promising, but the values, absolute values of the, for example, gray matter or white matter, they are a little bit off of the literature, but the overall, like, we have ground truth method, which is actually the big sequence with 30 images, and comparing to them, there is statistically significant improvement in the correlation between before optimization and after, and in other metrics like PSNR and etc. So let's move to the conclusions. So CEST MRF is a good approach, but we need further analysis and optimize all protocols. The best way to optimize is use analytical solutions, but it is not feasible for the clinical pulse sequences. Numerical optimization works, but there is some problems which still might be researched. In the future, I'm planning to incorporate deep learning. For example, we already showed that we can substitute our physical numerical simulator with a neural network. And the advantage is that it works 10 times faster when you have a dictionary with billions of parameters. It really helps because instead of like days of simulation, you can just spend hours. And also for optimization, there is a ways how to approximate analytical solutions with neural networks, etc. And also, if you're interested in this topic, very soon we were invited to submit a paper to Nature Protocols with details how to perform from the beginning to the end CEST MR experiment. And we will publish our code with examples with our simulator. So if you're interested, you can write me, and when it will be published, I will notify you. Yeah, and just a couple words about our lab. As you probably understood, we are focusing at the development molecular MRI methods for the diagnosis and therapy optimization. We mostly work with optimization deep learning, so it's mostly software work. We have access to the 7T scanner, 3T scanner. We just recently bought a cool NVIDIA A100. We have several other clusters, so you can check open positions at this website for PhD and postdocs. So thank you for your attention. I'm glad to answer to any of your questions.
Nikita, thank you very, very much. It was really interesting. So I think our colleagues can think about the questions, and I have several.
Okay.
So you just have told that you can share the simulator. So does this mean that you totally wrote, or the guys in the lab wrote their own simulator?
No, like
because I know that there is a like CEST PulseSec simulator, which
there is
available.
CEST PulseSec simulator, but it implemented in MATLAB and C++. And there is a problem that you cannot publish and open access some MATLAB thing because people need license. So the first my step was like implement the same. Like I took C++ code, wrote some interfaces for Python, and made it available for the multi-threading. So it actually PulseSec CEST the same is MATLAB but in Python. And now we can share it in open access. Yeah.
We just put us in the line of people who are waiting for it. It can be quite useful. Okay, so I wanted to ask a little bit more about the model which you use in this simulation. So do you consider the pulse profile there, RF pulse profile, or you just I mean then that when
you mean like the shape
yeah, excite the slice, the profile of the slice is not like rectangular, so the effect of magnetization transfer can be different in the center of the slice and in the border of the slice, so it can affect the result.
Yeah, in my case, we didn't consider that. Right now, it's simplest rectangular pulse, but in the more classical ways and APT weighted images and where you must acquire the full spectrum, it's shown that you're using some sync shapes or Gaussian pulses. It helps, like not only
No, I don't mean that what type of pulse do you use. I mean, do you consider the profile of the pulse in the modeling, in the modeling of the response of the pulse sequence? So when you use a block pulse, like rectangular pulse, so the Fourier transform of this pulse will be very long sync profile, yes? So this means that in the center of the slice, you will have like 90-degree pulse, but at the border of the slice and in the side lobes, there will be different angles for which you and the saturation will be different. So the saturation is not homogeneous inside the pulse profile. So I was just wondering, do you consider this modeling in the pulse selection, in the slice selection direction, or you just take it like as a one magnetization, which you just rotate?
So yeah, our simulation is like pixel wise.
Okay.
So yeah, there is no consideration of that.
Actually, in some
as far
block,
I understood. But maybe I
block
missed
simulations, yeah, in block simulations, not in block McConnell simulations, it's quite popular. It is quite, there are quite a lot of work where you can simulate taking into account this pulse profile. So this may be a good correction for the model actually, because it really affects the results. Just without magnetization transfer, just without considering magnetization transfer. And I assume that when you consider magnetization transfer, it will affect the result very much. So maybe you can take it into account.
I took a note. I will check it.
Okay.
Thank you.
And and you usually use single slice imaging, yes?
No, we are using our readout sequence the three-dimensional API, so we acquire the three-dimensional image at once.
Three-dimensional. Okay.
Yeah.
Okay, great. Okay, and you told us about SAR. So in case of human scanning, so what is the like total length or total duration of the pulse sequence, and which can be cannot affect very much the
Right
human
now,
body?
in all my sequences, I'm using saturation time 2.5 sequence and total with 13 pulses. And with duty cycle 0.5, it works okay. But when we increase to the, for example, 0.9, that's all, like it doesn't work on the scanner. So actually it must be, when optimization is performed, it must be put to the loss function to do not give to you solutions with SAR considerations. But right now, it's empirically founded the working sequence.
Okay, thanks. And one more question. You told that in the model, in the big model, which creates a lot these big dictionaries, you vary the frequency. But what is the reason for varying frequency when you create a dictionary for a particular metabolite?
Yeah, varying frequency is usable for MT because like MT has a broad like line, and if you want get better MT effect, it's useful to varying of frequency of irradiation. It gives better MT effect, better contrast.
Okay, sorry, I
so
lost
it's useful
connection.
not for CEST but for MT, which a bit related.
Okay, so by MT you mean magnetization
transfer, yeah.
Okay, great.
So
okay,
yeah.
Thank you very much. So colleagues, do you have any questions in addition to mine? Okay, maybe Zila or someone else who is working with very similar things? No? Okay, Nikita, so thank you very much. I think that probably if our colleagues will have questions, they will write you. Anna Kurkanin, you have your mic on, so do you want to ask a question? no, probably not. Okay then. So thank you very much, and I'm really glad that you are like taking the heritage from what you studied in your master's program. So I hope it is useful for you now. So and good luck.
Yeah, thank you for the invitation.
Okay. So the colleagues, so for today we are over. See you next week.